So thank you everybody for coming to GenomeConnect's April webinar about how to read your genetic test report. As a reminder, we did have a previous webinar in February about the importance of sharing health and genomic data. We also have another webinar, June 25th at 3 p.m. Eastern Time, where we will talk about the Brain Gene Registry, which is a GenomeConnect registry partner. All of our webinars are recorded and available on our YouTube playlist. The recording from the webinar in February is on YouTube and can be accessed using the QR code on the slide. Thank you to those who provided us with questions as part of your registration. We hope to answer a majority of these questions during this webinar. We will have time at the end of the webinar to take your questions. So if you do have questions during this webinar, please use the Q&A box located at the bottom of the screen. GenomeConnect has finally reached 10,000 participants, so we did want to take this time to thank all of you, as this would not be possible without your participation in GenomeConnect. This is today's webinar team. So my name is Alexis Morgan. I am one of the genetic counselors involved in GenomeConnect. I will pass it to Caitlin and Allison to introduce themselves. Caitlin.
Hi, everyone. Thank you all for joining. My name is Caitlin, and I'm also a genetic counselor with the GenomeConnect team, and I am here to help answer some questions and provide some technical support, which I obviously messed up already. So thank you all for your patience and rejoining on the Zoom link. Thanks.
And I'm Allison. I'm a genetic counseling assistant with the team, and I am just doing technology backup, so I'll be lurking in the background.
Right. So during today's webinar, we will start with an overview of different genetic concepts to help assist in our main discussion, which will be reviewing two types of genetic testing reports, a microarray report and a sequencing report. Individuals are made up of millions of cells, and those cells contain chromosomes. Chromosomes are structures that hold all of our DNA. Our DNA make up our genes, and each gene is an individual instruction for our body. Typically, people have 46 chromosomes total. Our chromosomes are numbered from biggest to smallest for the first 22 pairs. The first 22 pairs are called autosomes. Typically, a person gets one copy of each chromosome from each biological parent. The last set of chromosomes are our sex chromosomes. Typically, people either have two X chromosomes or one X and one Y chromosome. Wrapped up tightly in those chromosomes are our DNA. Our DNA provides instructions to make protein. And again, each instruction is a gene. The proteins play important roles throughout the body, and each protein has a different role within the body. Our DNA is made up of letters called nucleotides. There are 4 different nucleotides, A, T, G, and C, and our body reads these nucleotides to make proteins. The nucleotides are read in sets of 3. So a set of 3 nucleotides codes for one protein building block within the entire protein chain. These building blocks are called amino acids. So the example on the slide, the DNA sequence starts with A, T, G, which is the first set of nucleotides, and that codes for one protein building block of the entire protein chain. We all have changes in our DNA called variants. These are part of what makes us who we are. Some variants can affect our health, development, or risk for future health concerns. A person can have different types of genetic variants. So we can have variants at the chromosome level or at the gene level. Different types of genetic tests are able to detect different types of genetic variants. A karyotype looks at our chromosomes in big picture, while a microarray zooms in for some more detail, and sequencing zooms in even further for the most detail. You can think of these different genetic tests like a book set. A karyotype is the test that looks to see if you have every book in the set. A microarray looks at each book's chapters to see if all the chapters are there. And sequencing tests look at the sentences on the pages to see if there are any changes to sentences and letters. There are different types of sequence genetic testing, the main difference being how many genes the test is looking at. Whenever a genetic variant is found, a genetic testing laboratory tries to classify that variant's effect on health. Variants are classified into different buckets. We have benign, likely benign, uncertain or unclear, and pathogenic, likely pathogenic. Benign, likely benign means the genetic variant is not thought to impact gene function, therefore not expected to cause disease. Uncertain, unclear means there is not enough information about this variant to know its impact on health. We don't know if this can cause disease, and additional information over time may be able to help better understand these uncertain variants. And pathogenic, likely pathogenic means this genetic variant is known to impact gene function and contribute to disease. These types of variants may explain a person's symptoms, increase a person's chance to develop a certain condition, or increase the chance of having a child with a certain condition. When a genetic testing laboratory classifies a variant, they use many pieces of information to classify the variant into those buckets. For example, has this variant been seen in the general population? Was the variant inherited from a parent? What is the protein impact of this variant? Has the variant been observed in anyone with symptoms? And many more pieces go into how we classify variants. Depending on the information available, variants will then be classified into one of these buckets. As more information becomes available, again, a variant may change in classification. Any variant in any bucket has the potential to change. For example, a pathogenic variant might be downgraded to likely pathogenic, or an uncertain result might be upgraded to pathogenic, or even downgraded to benign, likely benign. In fact, we hope to reclassify these uncertain results into a more clear category. But not every variant will be reclassified, and there's no timeline for reclassification. Some uncertain results may be reclassified in a month, while others may still be uncertain after years. So now that we have reviewed some genetic concepts, we're going to review some genetic test report examples. First, we are going to start with a microarray report. This specific genetic test, again, looks at an individual's chromosomes. It's able to detect full missing or full extra chromosomes, which are called monosomy and trisomy, respectively. A microarray also looks within the chromosomes for some big or small missing or extra pieces of chromosome called deletions and duplications. Together, these chromosome variants are called copy number variants, or CNVs. So this example microarray report is meant to look similar to many microarray reports you may see, and we're going to review what each part of this report means. So starting with the result itself, if there is a chromosome variant identified, the result may say positive or uncertain. If there is no chromosome variant identified, it will say negative. This specific result says it's positive for a heterozygous 2.69 megabase duplication at 22q11.2. This is a lot of information, so we're going to break it down. Each chromosome has two arms, a short arm called the P arm at the top of the chromosome, and a long arm called the Q arm at the bottom of the chromosome. And each chromosome has specific banding patterns, which are the stripes you see throughout the chromosome. This report is stating there is a copy number variant, or CNV, on one of the 22nd chromosomes. Specifically, there's a duplication on the Q arm located at band 11.2. The report will also note the classification of the copy number variant. This specific report states that this is a pathogenic variant or a variant known to be associated with a condition. Another way you may see a copy number variant written is shown in the circle. ARR means this was a microarray. GRCH37 is stating the specific genomic database or genomic build that was used for this testing. You may also see different numbers for the genomic build. That is okay. Then it will state the chromosome and the banding pattern. So for this example, 22q11.2. The numbers in the parentheses after are giving the specific genomic coordinates the copy number variant is affecting. The first number is the starting point, and the last number is the ending point. Next, you will see an X and a number. This tells you how many times that specific copy number was seen. Typically, a person would have two of these regions, one on each chromosome. But in this example, this is a duplication. So we see this region 3 times. If it's typical for there to be 2 regions, but someone has a deletion, it would say ×1. Keep in mind, ×1 and 3 are not the only numbers you may see. Sometimes you may see ×0, ×2, ×4. The number can vary. Another thing you might see at the end of this section is the word MAT or PAT. This is only included if parental samples were also sent in as well. And this will tell you if the copy number variant was inherited from a biological parent. Reports will also have a results and interpretation section where they provide additional information about what type of copy number variant was identified, what genes are included in that chromosomal region, and what conditions might be caused by the copy number variant. This specific example states this duplication is consistent with a diagnosis of 22q11.2 duplication syndrome. If parental samples were provided for testing as well, the report will also note again if that specific copy number variant was identified from a parent. The specific example is paternally inherited, meaning this copy number variant was inherited from the individual's biological father. When a variant is inherited from a parent, that means the individual's parent also has the same variant that was identified in their child. The star in this picture represents a variant. It's seen in a parent, and it's also seen in the child, meaning it's an inherited variant. Paternal means it's inherited from the biological father, and maternal means it's inherited by the biological mother. Now that we have reviewed a microarray report, we will move on to reviewing how to read a sequencing report. A sequencing test looks at an individual's material in more information, more detail. It's looking at the gene level for any changes in the letter of the DNA, what we sometimes call spelling in the genes. These types of genetic changes are called sequence variants. Looking at the example on the slide, there is a DNA sequence, perhaps a portion of one gene. the top letters are the typical spelling for this gene. And in the patient sequence underneath, you can see a sequence variant where one of these letters changes to another letter. So in this example, where we would typically see a T nucleotide, we see a C nucleotide in the patient sequence. In this example sequence report, again, this is meant to look similar to most sequence reports, and we're going to review what each part of this means. These reports will mention the gene that was identified to have a sequence variant. So in this example, the gene is NF1. Sequence reports will also note if there is a disease or a genetic condition associated with that gene. In this example, the NF1 gene is associated with neurofibromatosis type 1, which is a genetic condition that impacts the skin. Individuals with this condition may have some dark spots called café au lait macules, freckling in the armpit or groin area, or bumps in the skin called neurofibromas. Not every gene will have a condition associated with it. Some genes may not have a condition or might be called genes of uncertain significance or candidate genes, meaning we don't understand the gene's relationship to disease. But based on some information, it could be tied to health, development, or disease. While some genes might not yet be tied to a condition, other genes might actually have multiple conditions associated with it. The variant information of the sequence report tells you exactly what genetic change or sequence variant was identified through testing. You will typically see a C dot and a P dot on the reports. The C dot is the DNA information, and the P dot is the protein information. In this example, the C dot or sequence variant is 8434C to T. The number 8434 is the nucleotide position, which means at the 8,434th letter of the NF1 gene, that nucleotide is typically a C, which you would call the reference nucleotide. But in this individual, there's a T at that position, which we would call the variant nucleotide. The P dot or protein description tells us what amino acid is typically in that position of the protein and how the protein is changed. So in the specific example on the report, this is what we call a nonsense variant. For this example, at this position, the typical nucleotide again is a C. In this individual, they have a T instead. In addition to just coding for amino acids in the protein building blocks, some groups of 3 nucleotides actually tell the body when the protein should stop. These are called stop codons. This specific change to the DNA sequence on this slide creates a stop instead of the typical arginine. Therefore, it stops the growth of the protein. So now this individual has a shortened protein chain, which can impact the protein's function in the body. So to summarize, a nonsense variant means there is a single letter change to the DNA sequence, and that single letter change changes the protein to stop early, therefore affecting the protein's function in the body. There are different types of sequence variants, and different sequence variants may have a different impact on the protein. This slide gives a quick description of different types of variants. We will review these variants in more detail in the upcoming slides. But we also have videos on our YouTube channel that review these types of sequence variants in some more detail. And those videos can be found also using the QR code on the slide. And I believe we are also putting the link in the chat as well. A missense variant is a DNA change that is often caused by a single letter change. This single letter change changes the amino acid in the protein. So in this example, the G, which is typical, was changed to an A in the DNA sequence. That G changing to an A changes the amino acid, which is typically serine, and it changes it to cysteine. This change in the amino acid may impact how the protein functions in the body. So to summarize, missense variance means there is a single DNA change that changes the amino acid in the protein, and that change in the protein may affect how the protein works in the body, but other times the protein function is not impacted by this change. Synonymous variants, also known as silent variants, are just that. The change to the DNA sequence does not cause a change to the amino acid. Therefore, it does not cause a change to the protein. So in the example on the slide, the sequence nucleotide, C, was changed to a T. But both TCC and TCT code for the same amino acid, serine. Since they both code for the same amino acid, it doesn't change the amino acid, therefore it doesn't change the protein, and the protein continues to function as expected. These types of variants are not expected to be associated with disease and are generally benign variants. So to summarize, a synonymous or silent variant means there is a change to the DNA sequence, but that change does not impact the protein, and these variants are benign and do not cause disease. Before, when we were talking about the microarray report, we talked about larger deletions and duplications within the chromosomes. The deletions and duplications that we're talking about now are different from those deletions and duplications. These sequence variants are smaller deletions and duplications within the DNA sequence. Adding or removing letters in the DNA sequence can result in two types of variants. The first variant is what we call a frame shift variant, which we will review now. The other is called an in-frame variant, which we will review next. As we talked about earlier, the body reads the DNA sequence in sets of 3. So insertions, duplications, or deletions of the DNA sequence, so any change in the DNA sequence, not in sets of 3, will shift how our body reads the remaining letters and can change the remaining amino acids, eventually resulting in an early stop signal for the protein. Since it is changing the reading frame, that's why we call it a frame shift variant. You can identify frame shift variants because they typically have FS in the description of the protein chain, or the P dot. The example on the slide is a duplication of 13 nucleotides, which is noted by the dup13 in the cDNA. This is not in a set of 3, so it's changed the reading frame of the DNA, therefore changing the protein, which is noted in the P dot. The protein changes and is shortened, so we see the FS denoting this is a frame shift variant. As I previously mentioned, adding or removing letters in the DNA sequence can result in two types of changes to the protein. The first change was a frame shift, and the second change is an in-frame variant. Insertions, duplications, or deletions of the DNA in sets of 3 does not shift how our body reads the remaining letters. since it is not changing the reading frame, it is called an in-frame variant. The example on the slide is a deletion of 3 nucleotides, which is denoted by the del AAC in the C dot. This is in a set of 3, so again, it does not change the reading frame of the DNA, but it was a deletion of one of the amino acids. So it did change the protein, which is noted in the P dot. The protein was changed and an amino acid was deleted, and that's noted by the del at the end of the P dot. With both frame shift variants and in-frame variants, we can have deletions and duplications. We can also have insertions of nucleotides, as well as something called indels, which is when a section of a sequence is deleted and replaced by a new sequence. These different types of variants can result in either frame shift variants or in-frame variants. The difference being between in-frame and frame shift variants is solely if the DNA change is in sets of 3 nucleotides or not. Our DNA have both exons and introns. Exons are the coding portions of the gene, and introns are the non-coding portions of the gene. The introns get cut out of the gene in order for the gene to produce the protein. Splicing is how the introns are cut out from the exons. They're the DNA sequence that help recognize the boundary between exons and introns for splicing to happen. So this can be compared to the drawings in school with the dotted lines showing you to cut here. A genetic variant in the DNA sequence that affects splicing is called a splice site variant. This type of variant disrupts the RNA splicing and can cause loss of exons or introns that don't get cut out, so they are included, which all can impact the protein. In this example, the body typically recognizes a splice site variant between G and G. But here, the second G is changed to a T, so the body is not able to read the splice site anymore, and therefore it's missed. On a report, this would show as plus or minus with a number after. So for example, +1, -1, +2, -2, etc. in the cDNA of the genetic test report. These types of variants have an impact on the protein by either shortening or lengthening the protein, which would then in turn impact the protein's function in the body. So now that we discussed a little bit about different types of variants, we will get back to the genetic test report. So next we have inheritance pattern. The inheritance pattern helps us understand how many copies of the specific gene need to be not working in order for someone to either have or be at risk for the specific genetic condition. Inheritance patterns don't refer to the specific patient, and they don't tell us if the variant was inherited from a parent. So in this example, NF1 is an autosomal dominant condition. For autosomal dominant conditions, that means we typically have 2 copies of the gene, and a pathogenic variant in only 1 copy of the gene is enough for somebody to have symptoms or features of the condition. Whereas autosomal recessive conditions means we typically have 2 copies of the gene, and there needs to be a pathogenic variant in both copies of the gene in order for somebody to have signs or symptoms of the condition. X-linked is another common type of inheritance pattern you may see on reports. For X-linked conditions, that means the gene that causes the condition is located on the X sex chromosome. Symptoms in people who have variants on their X chromosome can differ between those who have 2 X chromosomes and those who only have 1 X chromosome. This was not an extensive list of inheritance patterns, and we do discuss other less common inheritance patterns again in our YouTube videos. Zygosity tells us if the sequence variant was found on one or both copies of the gene. So this does tell us more about the specific patient. In this example, the patient, it's said to be heterozygous, which is used to describe when only one variant in a gene is found on one copy of the gene. Some other terms you might hear include single heterozygous, compound heterozygous, homozygous, and hemizygous. Instead of just heterozygous, some reports say single heterozygous. These two terms mean the same thing, that the variant was only in one copy of the gene. Compound heterozygous means that there are 2 different variants in the same gene, whereas homozygous means the same variant was found in both copies of the same gene. And hemizygous is when one variant was found in a gene, but that person only has one copy of that gene. This primarily includes the genes on the X chromosome in someone who only has one X chromosome. As mentioned before, inherited from is different from inheritance pattern. Inherited from tells us if this sequence variant came from one or both parents, or if the sequence variant was a brand new change in the individual. In this example, the sequence variant is de novo. De novo means the variant was not inherited from either parent and was a brand new change in this individual that occurred either in the egg or sperm or early on in development. Lastly, sequence reports will provide the variant classification. This tells you how the laboratory classifies this variant and how it may impact a person's health, and if this variant may cause a certain genetic condition. This example variant is classified as a variant of uncertain significance, or VUS. You may also see these noted as unclear or unknown significance. A VUS means the laboratory does not have enough information about the specific variant to know if it can impact health and cause the genetic condition associated with the gene. Sometimes these variants are changed or reclassified to benign, not disease-causing, or pathogenic disease-causing, as more information becomes available. But as a reminder, there is no timeline for reclassification. We would like to thank you all for joining today's webinar. We hope this has been helpful. Again, additional information on these topics can be found in our YouTube channel videos. And as a reminder, this webinar was recorded and will be available. At this time, I will take some questions using the Q&A box, which will also be recorded.
Hi, Alexis. Thank you so much for going through this. We have one question, and it's what about double hets or double heterozygotes? What does that mean?
So for somebody who is a double heterozygous, I'm assuming we are asking a little bit about that compound heterozygous. So you either have one change in one copy of the gene, and then you have a different change in that other copy of the gene.
Great, thank you. And then I'm gonna put in a few questions that I think we can answer together. And so one question is, can you tell me a little bit about truncated variants? And so what a truncated variant mean, and then a little bit about the difference between a missense and a nonsense variant.
Sure. Let me scroll back quickly to my missense and nonsense just to help with explaining. Okay. So here, with a nonsense variant, we have a C that was changed to a T. That C that changes it to a T changes the amino acid. So typically, the C would code for an arginine amino acid, but when that C is changed to a T, it actually changes that arginine to a stop. So a nonsense variant is a single change to the DNA sequence, and that single change changes the protein to a stop. So it affects the protein and it shortens the length of the protein. Versus a missense variant changes the protein from one amino acid to a different amino acid. So here, the typical variant is a G, and that G is changed to an A in the patient. When that G is changed to an A, it changes this amino acid from a serine to a cysteine, therefore possibly affecting the protein.
Thank you. And then to go on a little bit more about what a truncated variants are. So truncated variants usually are when there's a change in the code that results similar to, as Alexa was talking about, a stop codon. And so it shortens the protein. So the protein is not as long or doesn't have all the coding regions as it normally has. And so that's really what's called a truncated variant, which truncated is what is happening to the protein itself. So the type of variant is usually maybe a nonsense or a frame shift, or it's a type of variant that shortens the protein. So now the protein is not its normal length, and so it's unable to maybe complete its folding as it normally would. And then someone asked a good question about what is intergenic variants. And so those are variants that happened between genes. So we have our long strand of DNA as we showed, and not all of our DNA is coded into protein. The places that are coded into proteins are called exons. And then there are introns, and those are pieces of our DNA that are maybe between the genes or between the regions are expressed. And so sometimes these are called intronic variants, or these are variants that are between some of our genes. And another question we receive is, why aren't VUSs disclosed in carrier screening in autosomal recessive diseases? And so I'm sorry, I don't have a good answer for you for this one. Sometimes this is laboratory dependent. So sometimes this is based on a lab's preference if they are going to disclose VUSs or not. More often, we disclose VUSs in what we would call diagnostic testing. So when we are looking at individuals that have signs or symptoms, and maybe that VUS could be associated. So VUSs are not always disclosed in what we call autosomal recessive or carrier screening, because we're not looking to diagnose anyone. We're looking for definitive answers if someone is definitely a carrier for a condition. So they're looking to only report out more definitive classifications of variants.
And just to add on to that, sometimes if you do have an uncertain or a VUS result in carrier screening that's not reported, that VUS can change over time. So if the VUS changes to pathogenic and we now know that you're a carrier, usually a new report would be released and you would be notified.
And then we have a few additional questions that we received prior. So I just want to also address some of the questions we received from the audience when they registered. And does everyone with the variant in the same gene have the same symptoms?
So short answer, no. Longer answer, people with the same genetic variant in the same gene, even in the same family, can present differently. So some people may have every feature of a condition, whereas others may only have a few features of the condition. Also, genetic variants, they can impact people differently depending on what stage of life they are in. Some conditions might be more adult onset, so they typically don't impact children. But as children would get older, they may start to have risk for the condition and features in adulthood.
And is an uncertain result the same as a negative result?
An uncertain result and a negative result are different. So a negative result means there were no genetic variants identified in the test, or the variants were benign, likely benign, and not known to cause disease. Whereas an uncertain result is classified when there's a variant of uncertain significance. So again, that means that we don't currently know if the variant is associated with the disease or not, because there just is not enough information known or available about the variant.
And I'm going to answer the next question and two questions kind of at the same time. And it says, first question is, how do we know if the test reason and symptoms experienced are utilized to determine if a gene is a VUS or a pathogenic? And so this goes a lot to the ordering provider and some of the information that's included on the test requisition. So the laboratory will look at some of the clinical information that's provided to them when they are conducting the test and when they're evaluating the information that they have found and when they are trying to put all the pieces together. Also, as part of GenomeConnect, if someone does upload a copy of their genetic test results and provide answers to the health surveys, we submit that de-identified information to ClinVar. We include the genetic results and the symptoms, and then this information can be used in some of what we would call our curation efforts. So the National Institute of Health has helped funded the Clinical Genome Research Project, and as part of the work at ClinGen, as we see on the little icon, is to help evaluate genes of relationship to health. And then with an addition, we also look at variants to determine maybe those variants, are they associated with disease? Are they not associated with disease? And we do use some of the information that we receive from our GenomeConnect participants, including their genetic information and the health symptoms that they have included. And so that question, I would also encourage you to follow up with the ordering provider to see what information was captured on the test requisition. In addition, a question that we've received is, who should I contact if I have questions about my genetic test results or want to know if there is updated information? So I would encourage everyone to follow up with their provider who ordered the testing or a local genetics provider. And you can also try to maybe follow up with the laboratory that completed the testing itself. Alexis, would you add anything else?
If you don't have a genetics provider, or if you have moved and no longer have a genetics provider near you, you can always find a genetic counselor if you go to findageneticcounselor.nsgc.org. Okay. I believe that's all the questions that have come through. Is that correct, Caitlin?
Yes, those are all the questions. And I'm just going to put in the link to the Find the GC as you just mentioned.
Wonderful. So lastly, we do ask that you please fill out our post-webinar survey. The QR code on the slide can be used to access that survey, but we also will be sending a follow-up email tomorrow with the survey link as well. It's a quick 2 to 4 minute question, and the feedback is greatly appreciated. We thank you all again for attending today and hope you have a great night.
Thank you. And again, I am putting more links in the chat. So just give me 1 second. And this is for the survey. But as Alexis said, it's also available on the QR code. So thank you all for attending and have a great evening.